So it's my honor to introduce Dr. Kerry Byrne. She was my advisor both as an undergrad and as a graduate. And we did a lot of great work together. For my thesis, it was Astragalus applegate, but she's going to talk about the sagebrush steppe portions, and I did help a lot with some of these studies as well. I might even make a cameo appearance in there. I don't know. So yeah, without further ado, Dr. Kerry Byrne.
Okay, thank you for having me tonight. I'm just making sure I'm on. And I am a type of person who likes to pace around, walk around a lot. So I'm going to try and stay in one place so that people on Zoom can see me, I guess. It's better to stay here. Okay. All right, yeah, so thanks so much for having me. I am an associate professor at Cal Poly Humboldt, and I'm really excited to talk with you all tonight about some research that I've been doing in the sagebrush steppe related to seed banks. I find seed banks to be super exciting because you get to identify, like you grow plants and it's a bunch of unknowns, and it's kind of like a game to figure out everything that emerges from the seed bank, and you're dealing with tiny little seedlings. So it can be quite tricky to figure out what is in the seed bank. And so I found that as a botanist, I find it pretty fascinating. I think a lot of you would probably, if you've ever worked in a system where you're trying to identify plants where you don't really have all the features you need to identify them, that is what working with seed banks is like all the time. Okay, so I got to figure out how to, oh, here we go. Maybe I just clicked down here at the bottom. Oops. Great. Okay. So before I get started, I'm the only speaker up here tonight, but I wanted to acknowledge that this work has involved so many different people. I'm going to talk about two different projects tonight. The first one took place in southeastern Oregon, and I just wanted to acknowledge all the different people who've been involved, including, as Sean said, here's a picture of Sean a few years ago in the field. So thanks to all of these people for contributing to this project. And the second project is even larger. This slide, actually, I didn't make. My collaborator at Boise State University made this slide. And I had the opportunity to jump onto a very big project that she is the PI on. And I just did a little piece of the project looking at the seed bank at some sites in Idaho. Okay, so we'll start with an outline of what I hope to talk about tonight. So here's an itinerary. First, I'll talk about what is the definition of a soil seed bank and why is a seed bank important? I'll talk a little bit about seed-based restoration in drylands and defined drylands. And then I'll talk a little bit about my past and current research related to soil seed banks and some conclusions. So without further ado, what is the soil seed bank? So the soil seed bank is just the viable seeds that are either on the surface of the soil or buried in the soil. So even seeds that might be present in the litter, those would be considered part of the soil seed bank. And the seed bank usually is divided into two different parts: the transient seed bank and the persistent seed bank. Transient seeds are those that are viable in the seed bank for 1 year or less, whereas persistent seeds are viable more long-term in the seed bank. And I wanted to show this photo here because it has a good example of both a transient seed, or a species that creates transient seeds, and a species that creates very persistent seeds. This tiny little seedling up here is sagebrush, Artemisia tridentata, subspecies bassiana, so mountain sagebrush. And sagebrush is notorious for having transient seeds. They survive 1 year or less, in usually like 9 months or less in the seed bank, which you can imagine is problematic. There's a big fire that happens and it kills all the sagebrush. It makes it really hard for new sagebrush individuals to germinate. Next to it, in the same plot, in the same sample, this is common mullein, Verbascum thapsus, and it's a non-native species that has really persistent seeds. These seeds last upwards of 200 years in the soil seed bank. So I was up against a lot of that at my site in Idaho, where there must have been a few Verbascum thapsus individuals that produce prolific seeds. So in some of the plots, it's just like we had 200 or more common mullein plants germinate in just a small soil sample. So you get an idea of transient and persistent seeds. And then persistent seeds can be further defined by whether they're short-lived, they live 1 to 5 years versus long-term, like common mullein, which survives for 200 years or more. And each species has really different germination cues and dormancy mechanisms. I'll talk about that a little bit. In this upper photo here, the grass, this is a photo again for my project in Idaho. The grass that you see is all cheatgrass or Bromus tectorum, which is a really bad invasive species in the Great Basin. And it does not have a dormancy mechanism. Short story about cheatgrass. We collected these soil samples for this seed bank emergence study. We dried the soil completely, sieved it to remove rocks. And then we wanted to do a cold stratification, moist cold stratification treatment so that we could encourage species that do have dormancy to germinate. So we sprayed a little deionized water on our soil samples, put it in a refrigerator for 4 weeks with no light, right? It was in the dark. And when I took the samples out of the refrigerator, there were all these cheatgrass individuals that had germinated in the dark, just because as soon as they get a little bit of moisture, those seeds start to grow. So that's an example of a species without a dormancy mechanism. Another example is the species that Sean did his thesis work on, which is Applegate's milkvetch, or Astragalus applegatii. And this species it's a rare federally endangered species, and Sean has talked about it last year. I think he gave a talk at CMPS about it. But it has thick seed coats. And so that acts as a dormancy mechanism for Applegate's milkvetch. And the seeds have to be scarified so that water can get in, and then they will germinate. So they have a strong dormancy mechanism. Another example of a species that's pretty common in the Great Basin is dusty maidens. So dusty maidens has a 1-month cold, wet stratification or dormancy. It has to have moisture and cold temperatures for at least a month in order for the seeds to break dormancy. So all this to say is that species are really diverse, and seeds in the seed bank have all sorts of different mechanisms in order to achieve germination. So in dryland ecosystems like the Great Basin, where I spend a lot of my time doing research, species have, as I said, really different reproductive or life history strategies to cope with the environmental variability in that system. And so I just wanted to briefly touch on a few of these life history strategies that species in these areas have. On the left, we have, and also these botanical illustrations, not the one in the middle, but the other ones, are hot off the press. We commissioned an artist to make them for this project, and we just got them last week. So this is the first time anyone has seen them besides me, and I think they're so beautiful. So you'll see them scattered throughout this presentation. Anyway, okay, so we'll get back to life history strategies. On the left here, we have perennial shrubs and grasses. They tend to have a really resource conservative approach to growth and seed production. They typically have transient seeds, so those seeds really don't last for very long in the seed bank, but they're perennials, and so they the above ground plants themselves act as a long-term seed source. So just the plants being on the landscape, they're long-lived plants. In contrast, native annual forbs are a really interesting group, and they have really diverse bet hedging strategies. And what I mean by that is even within a given species, individual seeds have slightly different requirements for germination and emergence. And so if it's really, there's a good amount of rain for a good amount of time and it's cold enough or something, not all the seeds of that annual forb will germinate and grow. Some of them will and some of them won't. So it's really good at hedging your bets so that if in a system that has a lot of environmental variability like the Great Basin, not all the seeds germinate at once. They also tend to have really persistent long-lived seeds that can remain dormant for years in the seed bank. And then on the other extreme, we have exotic annual grasses. This is one of those beautiful illustrations that someone made for us. It's cheatgrass. And exotic annual grasses have a resource acquisitive growth strategy. So they tend to grow really quickly and get those resources out of the environment. And they tend to have really large seeds, and they don't have any vet hedging. As I said, with the cheatgrass in the refrigerator, as soon as it got just a little bit of moisture, all of the seeds started to grow. None of them stayed behind. So these seeds tend to just all start germinating with the onset of small rain events in the exotic annual grass life history strategy. Okay, so why are seed banks important? They provide insight into both the history of what was there on the landscape previously, maybe even the past 200 years or more, depending on how long the seeds can survive in the seed bank. And also they kind of predict the future of what's going to be there after the current species that are there die. Seed banks often show higher diversity than the corresponding above ground plant community. And they also tend to, I read an article recently about how seed banks tend to buffer disturbances. So maybe the above ground vegetation starts changing because of a disturbance, but the seed bank can kind of buffer that because it doesn't change as quickly. And then some species are only, not all the time, but in an entire lifetime, they may only be represented in the seed bank. So I'll talk about a species that I found in one of my seed bank studies that I was out at that site for 5 years, multiple times a summer. I never saw the species above ground, but then it came up 2 years in a row in the seed bank, which I thought was pretty interesting. And then finally, seed banks hopefully can be used as a resource for restoration. So drylands make up more than 40% of the Earth's terrestrial surface. And by definition, they're water limited systems. So we typically don't have or don't see very many trees in drylands. Instead, they're sort of the opposite of here. It's dominated by shrubs and grasslands. I think this visual will make sense to all of you. My husband will laugh that I'm telling it all to you. But when I was an undergrad, or even, I don't know, when I'm teaching, I tell folks to visualize upside down forests when they think of grasslands. So here in the Redwood Coast, we have like all of this biomass above ground, right, in the trees. And then the root systems, like they're cool, but they're not like ginormous. And then a grassland is kind of the opposite. There's all this biomass above ground. If you have a perennial grassland or perennial shrubland, there's a lot of root biomass below ground. So it's an upside down forest, right? There's not that much above ground that you see, but there's a lot below ground that you see, that you can't see. So because drylands are highly variable and have environmental limitation, they're particularly susceptible to degradation following disturbances like oil or gas extraction, fire, invasive species, or climate change. And in some cases, if there's a really big disturbance, natural regeneration or natural recovery may not be possible from the seed bank. And like think about, you know, a large wildfire, or in some restoration projects, like what's happening at Walda's, I think it's Walda Dunes, if I'm pronouncing that right, like north of Lanphier, the restoration that's happening there where they used large construction equipment to remove yellow bush lupin. Like there's not a lot of seeds left at those sites because they disturbed a very large area. And so in cases like that, land managers rely on seed-based restoration. And what I mean by seed-based restoration is when, here's an example. This example is from, it was a really big fire in Idaho, a pretty big famous fire whose name I can't remember at the moment. But after the fire, they went in with, this is farming implements. So they went in and reseeded using farming equipment after the fire. And that sort of large scale seed-based restoration is really popular. It's used a lot in the Western United States, but it's expensive and it's often ineffective. So for example, the USBLM spends about 50 million annually on purchasing and implementing seeding, but these seeding efforts are typically less than 10% effective. So even though we're spending a lot of money, there's a lot we still need to learn. And I don't want to make it sound all doom and gloom, because there's a lot of really cool researchers that are doing research on how we can make these seeding efforts more effective. But that's not really the research that I do. I think it's interesting, but I'm more focused on if we can use natural regeneration as an option. So I guess I kind of got ahead of myself, but a brief overview of my research. I'm really interested, or today what I'm going to talk about is how disturbances affect the seed bank. I'm going to talk specifically about drought as a disturbance, and also cheatgrass or species invasion as a disturbance, a large-scale disturbance. So how do those things affect the soil seed bank? And then my second question is if it's possible to use the seed bank for natural regeneration in some cases. So here's a map of the sagebrush steppe and Great Basin vegetation types in Western North America. And there we go. I'm going to talk about two different studies that I've been involved with. One is just over, it's not that far from here. It's just over the California border. It's in southeastern or southern Oregon on the Modoc Plateau. And the second project that I'll talk about is just outside of the Sawtooth Mountains on the Sawtooth National Forest in Idaho. I don't know, maybe we'll call that Central Idaho. Okay, so the first disturbance, as I said, I'm going to talk about is drought. And climate change is predicted to increase both the intensity and the duration of drought in drylands globally. And oops, I got to move this red thing. Okay. Climate change is predicted to have both direct and indirect impacts on the soil seed bank. So directly, climate change or drought may reduce seed production. If the plants aren't getting as much water, if it's hotter, they may produce fewer seeds. And it also might compromise some species bet hedging strategies. So if there's like a sort of a normal precipitation pattern that a site receives, and then it happens to be a really wet or maybe, the rain starts, but then a drought happens right after the precipitation starts. So all these plants or all these seeds might germinate because historically they were getting enough water to survive. But then if it's a really dry year, then all of those seeds that had germinated, a lot of them will die. So an indirect impact is that when soils are drier or hotter, it can actually kill the seeds or decrease the viability of the seed. So even if the plants are producing seeds, they may not last as long as they would have historically in the seed bank. And in drylands, shrubs are also really important. So shrubs are often considered nurse plants in drylands. Here's another one of the, oh, I don't have it up yet. Here's another one of those cool illustrations that's making its debut. This is supposed to be a sagebrush shrub. And when I say nurse plants, what I mean is that these shrubs are somehow helping the seeds that are below them and below the canopy of the shrub. In some ways that shrubs can act as nurse plants includes shrubs can increase soil moisture beneath their canopy from the shading, but also perhaps from hydraulic lift, which is when shrubs sometimes bring deep soil water up and then redeposit it beneath shrubs, which is really cool and was originally discovered in sagebrush. I already talked about shade. There can also be increased nutrient cycling beneath the shrubs. And so the seeds that germinate and emerge beneath the shrub canopy can have better nutrient status. The shrubs can also just offer different forms of protection. I read one paper a long time ago, but it always stuck with me, where in an area that was grazed, there's a lot higher diversity of plant species beneath the shrubs because the livestock couldn't eat them. So that's a sort of physical protection. They can also act as a wind or a water barrier. So that those are other ways that they can act as nurse plants. And so the prediction would be that if you're underneath the shrub, if we took some samples beneath the shrub canopy, we might find very different species, seeds for species than we would if we took samples not beneath the shrub canopy. And so our research questions for this project are first, how does severe drought affect the soil seed bank? Second, what is the importance of shrub microsites in terms of seed bank composition? And third, how similar is the above ground and below ground species composition? Today, I'm just going to talk about the first two questions and kind of ignore the third question. But this was work that my graduate student Allison Nunes completed at, then maybe it was Humboldt State. I don't think it was Cal Poly yet. But so this research has already been published, and I want to acknowledge this is really her work. Okay, so again, this project took place just over the California border, the Modoc Plateau in Southern Oregon. And let's see, so you can see this is a map of the field site. This is an ephemeral creek going through the field site, and there's two sets of plots, one on sort of the north side of the creek and one on the south side of the creek. These plots are really close together, but because of soil differences, there are different sagebrush species, which is pretty cool. They're like, the plots are 60 meters apart. They're super close, but they have really different plant communities because of the soil differences. And so on the left, we have the site that is dominated by low sagebrush or Artemisia arbuscula. This site has, you can't tell too much in this photo, but it has a lot shorter vegetation. And it also has a lot more bare ground than the site that's located on the other side of the ephemeral stream, which is dominated by silver sagebrush or Artemisia cana. So AA, Artemisia arbuscula, low sagebrush, AC, Artemisia cana, silver sagebrush. So they have differing amounts of bare ground, differing amounts of total vegetation, and the vegetation height is really different between these two sites. Both species don't have any other shrubs. Both sites don't have any other shrubs present. They have similar invasive grasses, annual brome species, and then bentonata or African wiregrass is a species that has invaded clayey soil, so areas that historically were not invaded by a lot of cheatgrass. Bentonata has come in, and so it's a really big problem in the Intermountain West, or it's becoming a much bigger problem in the Intermountain West, and it's pretty abundant at these sites. They have fairly similar perennial bunch grasses with some small differences, and then very different annual and perennial forb communities. So for this project, we did a drought manipulation. Where I guess I'm getting ahead of myself, but what we did is we set up 10 plots at each site, so on each side of the ephemeral stream, and then we randomly assigned the plots to either a drought treatment or an unmanipulated control that was just ambient rainfall. So there's 5 drought plots and 5 unmanipulated control plots at each site. And as I said, this was a drought manipulation. Here's a picture where you can see these drought shelters that my husband built. Shout out to Jason Yost, if he's in the audience. He's probably not, he worked a night shift last night. Anyway, so these drought manipulation shelters, they have corrugated, like clear greenhouse plastic strips going across the top. And it's a slanted roof. So when it rains, and those corrugated plastic strips that go across the top intercept about 41% of incoming ambient rainfall. So when it rains, 41% of that precipitation is channeled off the plot out of the area. And this 41% reduction in rainfall was based on long-term climate predictions for the site. Actually, I should clarify, that's not totally true. It was based on the historical precipitation record, and it was a 1 in 100 year event. So like an extreme drought for the region. So it's not predictions for, it's more of it's to simulate more of an extreme drought. So we left these rainfall shelters up year round from July 2016 to October 2020, but we collected our soil samples for Allison's seed bank study in May of 2019. Okay, so how we collected our soil samples, we used hole saws. I think Sean has some experience doing this as well. So, oh yeah, actually you came with me in 2019. You did. So Sean and I collected all the samples for this project. So we collected samples of seed bank samples, both, let's see, beneath sagebrush canopies and also in the surrounding inner space. So you can really see the amount of bare ground in this photo here. There's a lot of bare ground. This is the AA, the low sagebrush site. So we collected 40 samples per site. We brought them back to Cal Poly Humboldt, dried the samples, and then did a seed bank emergence study in the Cal Poly Humboldt greenhouse. So we didn't cold stratify the samples because we collected them right after wintertime. But we sowed the samples into trays and left them under well-watered conditions, left them in the greenhouse for 2 growing seasons. And then as seedlings started to emerge in each of these trays, we identified them, we recorded them, and then removed them from the trays so that we could make space for any other seeds to come up. So there was no competition. If we couldn't identify the seedlings, which is tricky, it's tricky to identify. I mean, how many of you have identified seedlings? It's very tricky. So if we couldn't identify the seedlings, we'd make notes about them, put them in a pot, and then grow them out in larger pots until they had identifiable features. Okay, so I'll talk briefly about some results. At the drier site with more bare ground and less vegetation, the AA site, we had 341 total individuals emerge, 10 total species emerged, and of those 10 species, 5 of them were only found in the seed bank. They weren't found in the above ground vegetation at the site. At the AC site, the site with more vegetation and no bare ground, there was a greater number of total individuals that emerged, a greater number of species that emerged, 19 species, and 9 of them were only found in the seed bank, not in the above ground vegetation. The majority of seedlings were exotic annual grasses, about 50%, and then annual forbs made up 40%, which was not super surprising. This is when I need to talk about, I was gonna say mimulus, but I guess I can't. Arianthri gutata, or yellow monkey flower. This is the species that came up in the seed bank 2 years in a row with 2 different sets of soil samples that I never saw above ground in 5 years of being at that site. And it's more of like a wetland. I think of it as a wetland or a seep species. And so it's really interesting that it was in the seed bank. And I don't think it was a fluke because it was 2 years in a row that it came up. Okay, there were few seedlings were native grasses, perennial forbs, and shrubs. And actually in this study, only 1 single sagebrush individual emerged, which was sad because that's like the keystone species of the sagebrush steppe. Some more quantitative results. Let me explain this to you really, or I'll take a second to explain this because we have 2 sets of graphs like this. On the x-axis, we have the effect size. And then here we have our AA site and our AC site. And we have a bunch of different response variables. What I want you to focus on are things that are in red, because the things that are in red are when there is a significant difference between the drought treatment and the control. Anything above the 0 line, this dotted 0 line, represents when the drought treatment has more of something than the control. And anything below the 0 line is when the drought treatment has less of something than the control. And so what I want to point out is, first of all, drought increased the amount of exotic annual grasses in the seed banks. There were more exotic annual grasses in the seed bank in the drought than in the control, significantly. The drought treatment had lower evenness and lower species diversity than the control plots at the AA site, the site that was had more bare ground. And we can see that there's only 1 significant difference at the AC site between the drought plots and the control plots. And that is in terms of exotic forbs. So drought actually decreased the amount of exotic forbs or non-native forbs at the AC site. Okay, so this increase in exotic grasses is mostly this 1 species, African wiregrass, that bad invasive that is becoming a big problem in the Intermountain West in clayey soils. And so that is bad news. It's really bad news. In terms of the decrease in exotic forbs under drought, that's a good thing, right? That there's fewer exotic forbs under drought at this site. However, it is largely due to this 1 species, Draba verna, which you can find here. It was like the plant of the day a few days ago in Lost Coast Outpost. So Draba verna, there's a finger for scale here. It is a minute, tiny little annual forb. So the fact that there's less of Draw Baverna in the seed bank under drought, I don't think it's that big of a deal. It's not like a great find. I mean, the species is not very important, so I downplay that. Okay, and now I want to talk about the seed bank response to shrubs. And so these are the same figures, we're looking at an effect size. And again, anything in red means a significant effect, and all the black things you can ignore because they're not different than 0. In this case, if the red line is a positive value over 0, it means that beneath shrubs, that value was higher than in the inner space. And so what you can see here is that at the AA site, that drier site with more bare ground, beneath shrub canopies, there were more native forbs than in the inner space. And also species diversity was higher beneath shrubs than it was in the inner space. But it only happened at that one site, the site that has a lot more bare ground. Because at the AC site, there are no significant differences. Shrubs didn't have any, there was no difference in the seeds between the shrub canopy and the shrub inner space.
Kerry, I'm tempted to slow you down. So for diversity, you're including the exotics species.
Yes,
as well.
diversity is all species.
And
Yeah.
then the top 4 things, that's the number of seedlings, not the diversity of that kind of plant, right? So native forbs is the number of seedlings of native forbs, regardless of what species they're in.
Yes. Yeah. Yeah. Versus diversity is calculated in a specific way. For diversity incorporates the number of individuals and yeah.
Yeah. So do you think that the exotics are changing the diversity statistic, or do you think the diversity statistic is reflective only of the natives? It could be either, but what do you think?
No, I think it's probably because there's a lot of invasive, especially well, here we're not looking at drought, we're like, well, yeah. So here there isn't their shrub microsite didn't affect we're looking at the shrub results, not the so there wasn't a difference in I see what you're saying. Like there wasn't a difference in invasive species beneath shrubs or in the inner space, right?
Let's say I only care about natives. Like if I only truly care about natives, then the exotics might have an effect on the natives, so I care about that, but I don't care about the diversity of exotics.
I think probably it's the native forbs that are driving the increase, because that's where, I mean, this was in terms of effect size, this was a pretty big one. Yeah. So that's a good thing.
I usually tell people not to interrupt.
That's okay. You can ask questions.
But the graph, I need to slow down.
Okay, so implications here. So what we found, remember, there are a lot more significant results at the AA site, that drier site with more bare ground and lower vegetation height. There was a lot more significant results due to drought and shrub cover there than there was at the other site, where the vegetation was more similar throughout the site. So an important implication is that seed banks of adjacent drylands may have different responses to drought and shrub microsites. I just said this before, even though I knew it was on my slide. I already just said this, but communities with bare ground and harsher edaphic features and lower productivity might have greater drought impacts. And shrubs seem to be really important microsites in the system that had a lot more bare ground. And so I hope these results are transferable to other drylands, but I don't know for sure. More research could see if it's true. And finally, also native forbs were resistant to 3 years of drought, but they really relied on those shrub microsites at the AA site. Also, I think this is a really important finding that exotic annual grass seeds increased under drought. And if this continues, then it could have potential negative consequences in terms of the fire return interval in this ecosystem. Historically, these low low sagebrush, steppe, these clay systems. They have a lot of bare ground in between the sagebrush individuals and the forbs. But if that bare ground fills in with a lot of litter from exotic annual grasses, it can carry a fire, and fires can and are becoming a lot more frequent in these systems. Okay. So stick with me. I'm going to talk about seed banks a little bit more. But we're going to take a little trip from Southern Oregon to the Sawtooth National Forest. And instead of talking about drought as a disturbance, we're going to talk about fire and invasive species as a disturbance. And I need to tell a little background story about how I started doing this work. For my 40th birthday, my husband bought me a flight to visit my best friend from graduate school, and she's a professor at Boise State University. And so we planned on like going on a backpacking trip. I don't know, we always joke about having a spa day, but we've never had a spa day. But then she got this really great Western SARE, which is Sustainable Agriculture Research and Education grants. And so I was still going to go out and visit her, but instead of having a spa day or going on a backpacking trip with her, I helped her set up her big long-term project, which was equally as fun. But while we were out there, this is a picture from us out there. It's a very steep, this is in the Sawtooth National Forest, higher elevation, sagebrush steppe. It's mountain big sagebrush is the subspecies of Artemisia tridentata there. It's very steep. You can't really tell from this photo, but it is an incredibly steep site. And all this kind of red in the understory, that's all cheatgrass. So she set up her plots, or I helped her set up her plots in really highly invaded cheatgrass areas on purpose. But I wanted to make a point, you know, this was sort of when we were out there working together, we talked about how great it would be to work together, and we hatched a plan for me to maybe do my sabbatical working with her in Idaho. We wrote a grant proposal and it was funded, which was amazing. But it wasn't just because we wanted to work together that we decided to do this project. I wanted to show a quote from a sheep producer in the area that's a collaborator on the project. So he says, this is Riley here on the left at the Western Wool Growers Conference, a couple in 2023, I guess. So a quote from him is, he said, "I see a lot of potential to use sheep as a tool to help improve overall vegetation health. On my grazing allotments, I've seen how sheep can help clean up areas with cheatgrass and make space for the native perennials to come back." So anecdotally, he's seen that in areas where he's a sheep producer, so he has a lot of sheep in grazing allotments on the Sawtooth National Forest. And he's seen that if his sheep, he uses the words clean up, sheep can clean up an area. And then he sees the native perennials come back. And that sentiment is shared by a lot of producers, a lot of livestock producers, but especially sheep producers in Idaho and elsewhere. And the question I have is, I mean, the sheep are eating the cheatgrass, but it's not as if Riley's going out there and planting seeds, right? So if he's seen the perennials come back after sheep have removed cheatgrass, well, they must be in the soil seed bank, right? And so that was really the impetus for our project. And it got us thinking about if you could use the seed bank for natural regeneration after a disturbance. And this project is still definitely a work in progress. So I don't have any totally official results to share with you yet, but I'll share what I have. So cheatgrass and other exotic annuals continue to expand in the Western United States, despite ranchers and producers spending billions of dollars a year on weed abatement. Targeted grazing, which has a lot of different definitions, but I'm going to define targeted grazing as high intensity, short duration grazing. So targeted grazing can be used as a vegetation management tool if it's implemented when the sort of desirable perennial vegetation or perennial grasses are dormant. And so the objectives for our project, for my sabbatical project, were to quantify differences in species composition between the seed bank and above ground vegetation within this larger targeted grazing project that my my colleague and good friend are conducting. And then also our second objective was to look across a gradient of cheatgrass invasion from plots that have very little cheatgrass above ground to plots that are like 99% cheatgrass, and see the effects that that invasion has on the soil seed bank. Okay, so as I mentioned, this project is taking place on the Sawtooth National Forest, which is located here in Idaho. And it's in a really, I'm trying to think of some background information that I want to provide you with. These grazing, it takes place in 2 grazing allotments that have historically been grazed by sheep. And the reason why they're grazed by sheep is because it's so steep. Cows and steer cannot go on these slopes. They're so steep, versus sheep are pretty adept, and they're much smaller animals. And so they can go in these steep areas that you could not have cows on. Okay, so Kelly, my friend Kelly, has set up four different grazing treatments. One is fall targeted grazing. One is fall and spring targeted grazing. One is spring targeted grazing. And then the control is the historical treatment, which is summer once over grazing, which is what these producers that run their sheep on these grazing allotments, that's historically what is done. You just, over the summertime, you bring your sheep out and they do a light one over. But what she's doing in this project and what the producers are doing is really intense, short duration, but like a lot of livestock or a lot of sheep in a small area. And I have a photo that I'll show you in a second. So there's those 4 grazing treatments replicated 4 times across 2 different grazing allotments, Deer Creek and Lime Kiln. And each summer, herders take about 1,000 ewes to each treatment plot per year, and that's happening across 3 years. And hopefully it'll happen more long-term, because we want to look, or she wants to look more long-term, what will happen. So in 2023, for this project, like my sabbatical work, we established, I think I mentioned, all of her plots are set up in areas that have a lot of cheatgrass. But what we did is we set up 8 additional plots that are shown in gray here. And those are in areas that have really low, they're close to the areas with high cheatgrass geographically, but they have very low cheatgrass cover. Okay, so here's just a photo, a great high-res image that kind of shows, this is the fall grazing treatment. And you can see these are all sheep. And if you look really closely, and they're all together, if you look really closely, there's some cattle, some livestock dogs that are out on the landscape, these little black dots. And then this is a herder right here. So they're this is, traditional sheep production. Most of the herders are Peruvian in this area. And they are out with the sheep. They're not using any fences. It's, the livestock dogs and the herders that are out with the sheep all the time, keeping them together. And it's really, so Kelly, my collaborator, really has to work closely with all of them. And it's pretty cool. Okay, so our methods. This is a photo of me 2 summers ago collecting above ground plant species composition data in one of the plots. So we collect above ground plant species composition in all 40 of the plots. We collected, I believe, 30 soil cores from each plot across all 40 plots. We dried those soil cores and then sieved them to remove any rocks or large pieces of litter. And then we did a cold, wet stratification treatment, where I already mentioned it, when the cheatgrass germinated in the refrigerator. So we moistened all of the samples and put them in the refrigerator at 2 degrees Celsius for 4 weeks. And that was to encourage breaking dormancy in a lot of these annual forb species that are present in these systems. And then we did another seed bank emergence study at the Boise State University greenhouse, which is just concluding soon. So all of these trays are gone, but we have a few tough perennial grasses that have been there for like 2 years now, and they have no inflorescence, and they're really hard to ID. So if you have a tip for getting native grasses to flower so I can identify them, let me know. Come talk to me after this. Okay, so some results. This system, the Mountain Big Sage system, is known for its species diversity. It's higher elevation, it's cooler, and it does have really high diversity. Above ground, we found 114 unique species, and in the seed bank, we found 50 unique species. Some of those are still unknown. Statistically, like a higher proportion of the seed bank was introduced species and annual species than in the above ground plant community. These findings were not that surprising. They were expected. What was interesting, and I'm not going to show you any figures about these data, but I am going to show you my really beautiful botanical illustrations, is there were a lot of native species that emerged in the seed bank as well, a surprising number of native species. So we have, I think our second most abundant grass after cheatgrass is sandberg bluegrass, which is a really important perennial bunch grass in this system. Came up in a lot of the plots, over 20% of the plots. Mountain hollyhock is another, it's a really nice perennial forb. I think sheep really like it. It came up in the seed bank. Blue bunch wheatgrass is really abundant above ground at this site. And we think it came up in the seed bank, but it has no inflorescences yet. And so I'm not totally sure about that, but we might do some genetic work to figure out what it is. And then arrowleaf balsam root is such a charismatic species. I mean, how can you not like this species? It did not come up in the seed bank at all, but it was really abundant above ground. So I included a photo or a picture of it. And I have some ideas why it maybe didn't come up in the seed bank, but I was bummed that it didn't. Okay, now I will show you a couple figures. Here we're looking at seedling density. So number of seeds per meter squared in the seed bank of cheatgrass, mountain sagebrush, and sandberg bluegrass. So cheatgrass came up, unsurprisingly, it came up in almost all of the plots. And it had a really high density, mean density, so 623 seedlings per meter squared. So there's a lot of cheatgrass. But remember, most of this study was set up in high cheatgrass invaded sites. Sagebrush came up in 22.5% of the plots, which actually I was really pleased with. The amount of sagebrush, or the number of sagebrush seedlings, was much higher in this project than in the other ones that I've done in Oregon. And in the plots where it came up, the average density was 29 seedlings per meter squared. But only, that's just in the plots that it came up. That's how many individuals came up per meter squared. And then finally, the sandberg bluegrass came up in 25% of the plots. And in the plots where it came up, it had pretty high density as well. 37 seedlings per meter squared. So I think all of this, I mean, cheatgrass, I'm not surprised that it's there, and I'm not surprised that it's abundant. But I think it's really promising that we have these important native species that are used for restoration that came up in the seed bank. Here we're looking at the relationship between above ground cover of a species and the density of that species in the seed bank. And so cheatgrass, unsurprisingly, the more cheatgrass there was above ground, the more cheatgrass there was in the seed bank. So there's a strong positive correlation between above ground cover of cheatgrass and cheatgrass density in the seed bank. Kind of surprisingly, the density of native perennial species in the seed bank was not correlated with their above ground cover, like across the board, except for sagebrush, which did have a marginally significant positive relationship. So the more above ground cover of sagebrush there was, the more there was in the seed bank. So for most species, you can't really predict what's in the seed bank based on their above ground cover at that time. Some implications from this project, which, as I said, is still ongoing. I thought it was really promising, or we think it's really promising, that native species emerged across a broad range of cheatgrass cover, which suggests that you shouldn't just look at what the above ground cover in the system. It could be pretty degraded, There could be a lot of cheatgrass or some other invasive species, but that doesn't necessarily mean that that there aren't some native species in the seed bank. And common sagebrush restoration species like blue bunch wheatgrass, sandberg bluegrass, and sagebrush are all capable of natural regeneration. So this might be an economically practical strategy for aiding in high elevation sagebrush restoration under certain circumstances. I'm not saying that like we can always rely on the seed bank, but I think there are certain circumstances where we might be able to rely on the seed bank. And so I think, you know, this study is ongoing, and I hope to do a I really hope that we can get funding to do a follow-up seed bank study to see after several years of targeted grazing by the sheep, if the seed bank is affected, positively or negatively. Are they getting rid of the cheatgrass, or are they nailing all the native species as well? But I think that the idea here is you could couple removal of some sort, like in this case, we're using sheep, but some other removal process, with natural regeneration to achieve some restoration objectives under certain circumstances. Obviously, if the entire site is nuked, you're not going to be able to rely on natural regeneration. Like if there's a major fire or you've removed all of the topsoil, you're not going to have any seeds really deep. Okay, I'm kind of getting ahead of myself, but I will end with one conclusion side. So I think restoring degraded ecosystems is one of the grand challenges of the 21st century. And the research that I'm doing and that a lot of people are doing about seed-based restoration or natural regeneration is really timely. In fact, the United Nations has declared this century the decade on ecosystem restoration. And so from their website, they say, there has never been a more urgent need to revive damaged ecosystems than now. I think probably a lot of us in this room probably agree with that. But you know, the point that I want to make is that a multi-pronged approach is needed. I don't think we can keep doing what we're doing, where we're throwing a bunch of money into seeding and hoping for the best when we're getting less than a 10% success rate. So there's a lot of folks that are doing other sorts of research, thinking about different treatments you can do to the seeds, which is not the type of work I do, but you can, you know, like put things on the seeds to make them not germinate as quickly, or thinking about different species you might use for restoration. But another idea is to think about what's already out there on the site. And so I think depending on the size and the intensity of the disturbance, the seed bank might be a good viable resource for restoration, for passive restoration. So nature-based solutions like this idea of combining sheep grazing, targeted grazing as a vegetation management tool prior to helping restoration occur more naturally, I think holds a lot of promise. But as everyone, concludes, a lot more studies are needed. We need more studies on like germination cues for different species. We need more studies to know like what's in the seed bank in different systems. It's a lot of work to conduct seed bank studies. So I think they're growing in popularity, but there's definitely room for improvement. And just in general, we need a lot more research about restoration so we can get past that 10% success rate. Okay, so with that, I'd like to thank you all for listening, acknowledge some of my funding, and then I'm happy to take, oh, I forgot, I'm happy to take questions, but also I brought some seeds to show you. So after the talk, I brought a couple different species just so you could see like the variation in the size and the types of seeds. So I brought a native perennial grass, I brought sagebrush, and a federally endangered plant species, applicates milk vetch. Okay, so time for questions if you have any. Oh, thanks. Maybe I'll end slideshow.
Sure.
And then,
I can do it.
okay, sorry about that. Oh, there is maybe, oh, this is a comment from someone who saw a plant, but I can take, sure, well, if you have a question?
You mentioned something about fire that you used, or
Oh, I haven't done anything with fire. At that site in Idaho, there was a fire. I don't know if I mentioned that, but there was a fire a while ago, and that's why the cheatgrass all invaded, was after the fire. I don't know. I don't think it was. There's not that much fuel there because it's not it's not a forested system, but I don't think it was a super intense fire, but I unfortunately don't know all of the history since I know it's only tangentially involved in the project with the seeds. Yeah, sorry. Iliamna rivularis. I believe I got that one right. It's definitely Iliamna. Yeah. Pardon? Cool. Yeah, I know. It's like easy to identify because I think they have stellate shaped hairs and they look very mallowy.
I was gonna ask if you have or familiarity or like context with the kinds of dispersal or seeding based restoration methods that are out there, sort of beyond what the BLM implements. And I'm asking just because I know that BLM lands tend to be managed more for recreational purposes and because there is so much like resource extraction activities that still happen on a lot of BLM land, it doesn't surprise me that their tactics would probably be pretty slapdash, you know, and that is kind of more about band-aids than like full on habitat.
Yeah, I think it also depends on the size of the project because you can have a lot higher success rate if you're using plugs, like individual plugs, instead of like the larger the plants are that you plant, the more likely they are to survive. But it's hugely more expensive to use plugs than it is to use seed and blast seed across the landscape. And so I wouldn't blame the BLM because they're dealing with huge areas versus if you're dealing with a very small area, you can be more successful because it's gonna cost more per square meter, but you're only gonna, if it's a small project, then it still can be affordable. But there's a huge range of the types, there's a big push for using more locally sourced seeds in projects. But there can be an argument for not using locally sourced seeds as well if you want to think about climate adaptation, right? Like maybe should you be planting what is already there, like the local genetics, or should you be planning for the future where the climate's going to be hotter? So there's like it's a really big area and there's lots of different techniques that are used. But I don't know if that really answered your question, but just I think it's just like there's a size issue. If you're trying to restore someplace that's huge, yeah, you're going to use seeding. But if it's a smaller situation, then you might be able to do more weeding and plant plugs and things like that, or rely on what's in the seed bank. Yeah. Yeah. I don't know. There's lots of questions. I guess Sean, you can.
Sure.
Okay.
I know nothing of the details, but I know that when the fungi people want to know what's there in the soil, they do the DNA thing.
Yes.
That's
That's a great question. I've talked with people about it because I wonder. So should I repeat the questions for Zoom? Okay. So the question is related to whether or not you could figure out what's in the seed bank by doing like a DNA analysis, like an eDNA analysis. And I have not seen a paper where someone has done that, but I don't think what I wonder about is figuring out what is actually there versus like still there and viable, or could there be genetic information in the soil that's not like it's from an above ground plant like root that broke off or something. So I wonder, I haven't seen anyone successfully do an eDNA project looking at the seed bank, but I feel like it's going to happen eventually. And I was talking, Karen Hull, she's a professor at UC Santa Cruz. I was just talking with her at a conference recently, and it seems like it's not that far off. Yeah.
You mentioned it's about a 10% seeding rates.
Or success rate, yeah.
Success rates. The
So is your question like what the viable, like if they're broadcast seeding, what percentage of those seeds were viable to begin with? Usually, at least with the seed that I bought from a company for another project, it said, they test it and it says like on the tag, 97.5% viable seed or something like that. But I don't know if that's really your question.
Yeah, I guess what is the viable percentage of sagebrush and that's
Yeah, good question.
pretty big for the environment.
And the problem with sagebrush is how do you collect it? And it's a transient seed. You collect it and then you have to plant it the same year, basically. Yeah. Was there,
So just, better comment. So recently, well, for the invasive, you're trying to get rid of the fence, really love the app. Will come back big time. They need a second floor permits to get out, and then it radical change. So anyway, just to not open the door. I'm not sure how fire would happen. But probably it's not to get rid.
yeah, I think it depends on the system and what you're doing restoration for. Sagebrush doesn't resprout after fire.
Right.
Yeah, but other species do resprout after fire and have a historical fire return interval that's much shorter than a lot of the different sagebrush systems. Yeah, perhaps, yeah, yeah. How are we doing on time for?
I think it's actually kind of nice for people to come up and talk with you later, but if you want to take a few more questions, you could.
What time is it? 8:42. Does one person have one more burning question they want to ask, but I can, okay, and I'm also happy. I have my seeds up here to show folks too. Gosh, that's kind of a loaded question. The question was about if these systems were historically grazed. So there are native grazers in these systems. The intensity is probably, or the density is probably a lot lower. Bison tended to not go like west of the Rockies. So really when we consider like the really grazing adapted species, we think of the Great Plains in North America. Because that's where all the bison were. So there was a really intense grazing pressure. And so that evolutionary history of grazing, but less so, like there are native grazers, but less long evolutionary history of intense grazing in the Intermountain West. Yeah. Okay, well, thank you all for your time. I'm happy to take more questions in the front and also show you my seeds. Want to see seeds?